Bismillahirrahmanirrahim. Assalamu alaikum dear students. I am Razia Bashir from University of Education, Lower Mall Campus, Lahore. I am co-teaching the subject of General Biochemistry with Dr. Muhammad Tariq. The topic which we are going to discuss today is Kreb Cycle, which is named after its discoverer. It is also known as tricarboxylic acid cycle because the first product in this cycle is tricarboxylic acid cycle, which is known as the citrate. So this cycle is also known as citric acid cycle. In our previous lecture, we have learned about the fermentation. As the end product of glycolysis is pyruvate, so there are different fates of pyruvate. It can be processed through the fermentation, which is anaerobic fate of the pyruvate, while the aerobic fate of pyruvate leads it to the Kreb cycle. This Kreb cycle takes place in mitochondria. So in aerobic organisms, glucose and other sugars, fatty acids, and most amino acids are ultimately oxidized to carbon dioxide and water via the citric acid cycle or the Kreb cycle and the respiratory chain. All these processes, they take place in mitochondria. Before entering the citric acid cycle, the carbon skeletons of sugar and fatty acids are degraded to the acetyl group of acetyl coenzyme A, and this process takes place in cytoplasm. The acetyl coenzyme A is the only form in which the cycle accepts most of its fuel input. It means whether these are fatty acids, these are amino acids, or these are sugars. First, they have to be converted into acetyl coenzyme A in cytosol. This acetyl coenzyme A is then led to the Kreb cycle, for which it is entered through the membrane of mitochondria. Many amino acid carbons also enter the cycle this way, although several amino acids are degraded to other cycle intermediates. However, amino acids can enter into the Kreb cycle at different stages. Before starting a Kreb cycle, we need to generate acetyl coenzyme A. So pyruvate is oxidized to acetyl coenzyme A and carbon dioxide by the enzyme pyruvate dehydrogenase complex. It is an oxidative decarboxylation which takes place in cytosol of a cell. So here in this picture you can see the overall reaction which is catalyzed by the pyruvate dehydrogenase complex. It is actually the 5 coenzymes participating in this reaction, and the 3 enzymes that make up the enzyme complex are later discussed. You see, pyruvate is a monocarboxylic acid. In the presence of this enzyme complex, it is converted into acetyl coenzyme A, and in this process an NAD is also reduced to NADH. Kreb cycle involves following main steps. The first is formation of citrate. Second is the formation of isocitrate via cis-aconitate. Then oxidation of isocitrate to alpha ketoglutarate and carbon dioxide. Oxidation of alpha ketoglutarate to succinyl coenzyme A with the release of a carbon dioxide molecule. Then oxidation of succinate to fumarate, hydration of fumarate to malate, and finally the oxidation of malate to oxaloacetate. This oxaloacetate, upon the provision of acetyl coenzyme A, is ready to begin another Kreb cycle. Let's have an overall picture of the reactions that takes place in Krebs cycle. Starting with the acetyl coenzyme A, which is formed in the cytosol. This acetyl coenzyme A through the mitochondrial membrane enters into the mitochondrial matrix where the oxaloacetate is present. This oxaloacetate combines with the acetyl coenzyme A in the presence of enzyme citrate synthase. And the first product is formed, which is the citric acid or citrate. It is a tricarboxylic acid, so the name of this cycle is also the tricarboxylic acid or TCA cycle. In this process, a water molecule is also utilized for conversion of coenzyme A. The second step is conversion of citrate to cis-aconitase in a reversible process in the presence of enzyme aconitase. In this process, a water molecule is released, so it is a dehydration reaction. The next step is conversion of cis-aconitase to isocitrate in a reversible reaction utilizing a water molecule in the presence of enzyme aconitase. In the next step, this isocitrate in the presence of enzyme isocitrate dehydrogenase is converted into alpha ketoglutarate. This is a step which is actually the oxidative decarboxylation, in which a carbon dioxide molecule is released from the overall carbon chain of a acid. In the next step, the alpha ketoglutarate is converted into succinyl coenzyme A with the release of a water molecule using coenzyme A, and the overall enzyme in this synthesis reaction is alpha ketoglutarate dehydrogenase complex. In the next step, succinyl coenzyme A is converted into succinate by the release of coenzyme A. In this process, the GTP molecule is converted into GTP, which is equivalent to the energy of an ATP molecule. So this is the step where basically energy is produced in overall Krebs cycle. The enzyme which is used in this reaction is succinyl-CoA synthetase, because here it is involved the synthesis of coenzyme A. In this process, as GTP is produced, so this is a substrate level phosphorylation. In the next step, succinate is converted into fumarate through a dehydrogenation reaction, and in this process an FADH2 is also reduced. The enzyme which is used in the process is succinate dehydrogenase. The next step is conversion of fumarate to malate in the presence of enzyme fumarase through a hydration reaction using a water molecule. And the next step is conversion of malate to oxaloacetate. It is also a reversible reaction in the presence of malate dehydrogenase. In this reaction, actually the overall reaction is dehydrogenation. The final product of this cycle is oxaloacetate in the mitochondria. This oxaloacetate, upon provision of one more acetyl coenzyme A, is ready to begin another Krebs cycle. Here you see in the Krebs cycle we have produced GTP from ADP, We have also reduced FADH2 and NADH. This is the end of our today's discussion. Thank you, dear students. If you have any question, you can ask your questions through the WhatsApp group or in Google Classroom.